Hello students, in the previous video I started with dark phase. We studied what is dark phase, and now in this video I am going to start with C3 cycle, that is Calvin cycle. Okay, C3 cycle or C3 pathway. The C3 pathway is also called as Calvin cycle because the C3 pathway has been explained by Calvin and his co-workers by conducting experiments on chlorella and synthesise. Hence it is also called as Calvin cycle. The C3 pathway or C3 cycle is also called as RPP pathway, that is reductive pentose phosphate pathway. Reductive pentose phosphate pathway. Phosphate pathway. Okay, right. It is also called as PCR cycle, that is photosynthetic carbon reduction cycle, where carbon dioxide is reduced into carbohydrate through the process of photosynthesis. So, photosynthetic carbon reduction cycle. Carbon reduction cycle. Okay, now let's see how this Calvin conducted experiments on chlorella and synthesise. So, Calvin collected vigorously growing chlorella, that is unicellular green alga, and to this chlorella he provided carbon dioxide. Carbon dioxide where the carbon present in this carbon dioxide is radioactive carbon, that is C14. Normal carbon means C12, but the carbon dioxide which is provided to this chlorella is with radioactive carbon. So after providing this carbon dioxide, radioactive carbon dioxide, he observed the fate of this carbon dioxide. Fate of this carbon dioxide is nothing but where this carbon dioxide is fixed and into which compound it is fixed, resulting in the formation of what type of photosynthetic product. Okay, so to identify the fate of this carbon dioxide, he used two techniques. One is 2D chromatography. And the other technique is autoradiography. So with these two techniques, he studied that the carbon dioxide fixation product which is formed is 3-PGA. After fixation of this carbon dioxide, chlorella formed a compound, that is 3-carbon compound, which is 3-PGA. 3-carbon compound. As the first carbon dioxide fixation product is 3-carbon compound, this cycle is called as C3 cycle. Okay, now this 3-PGA is with radioactive carbon in it, and hence he found that this carbon dioxide is fixed into a compound which has become 3-PGA, and hence he thought that carbon dioxide is fixed into 2-carbon compound, and hence as the carbon dioxide is 1 carbon and as it is fixed into 2-carbon compound, the resultant product, carboxylation product, is 3-PGA, 3-phosphoglyceric acid, which is 3-carbon compound. That is about the earlier view. But now scientists found that this first carbon dioxide is fixed into RUBP. Now let's study the brief details of this Calvin cycle in brief. Then let's study the details of this Calvin cycle in detail. Okay, let's start with the details of Calvin cycle. So earlier it was thought that carbon dioxide is fixed into 2-carbon compound, and hence the first carbon dioxide fixation product, carboxylation product, is 3-carbon compound, that is 3-PGA. But now scientists found that the carbon dioxide is fixed into 5-carbon compound but not into 2-carbon compound. So carbon dioxide is fixed into 5-carbon compound, that is RUBP, which is ribulose 1,5-bisphosphate. It consists of ketone group, hence it is ketone sugar. Okay, so carbon dioxide is fixed into this RUBP, that is 5-carbon compound, by the activity of an enzyme Rubisco, resulting in the formation of 2 molecules of 3-PGA, 3-phosphoglyceric acid. So here this Rubisco is a dual enzyme. It functions as both carboxylase and oxygenase. Rubisco, RUBP carboxylase and oxygenase. So for the activity of this enzyme, 3 factors are required. They are light is required, carbon dioxide is required, and even Mg+2. Magnesium is also required for the activity of this enzyme Rubisco. So this Rubisco is involved in the fixation of carbon dioxide into RUBP. That step is called as carboxylation, which results in the formation of 2 molecules of 3-PGA. So this RUBP is 5 carbon compound, carbon dioxide is 1 carbon compound, resulting in the formation of 2 molecules of 3 carbon compound, 3-phosphoglyceric acid. Now this 3-phosphoglyceric acid undergoes next step of this Calvin cycle. Calvin cycle involves 3 steps. They are carboxylation, reduction and regeneration. Now it enters into next step, that is reduction. So this reduction occurs by 2 steps. In the first step, this 3-PGA undergoes phosphorylation into 2 molecules of 1,3-bis-phosphoglyceric acid. 1,3-bis-PGA. So here for this phosphorylation, phosphate is donated by ATP. Phosphate is donated by ATP and that phosphate is added at first position. Already phosphate is present at third position and new phosphate is added at first position, and hence this 3-phosphoglyceric acid has become 1,3-bis-phosphoglyceric acid. Okay, so this is the first step of reduction, that is phosphorylation, during which ATP is consumed. Now coming to second step of reduction. In the second step of reduction, this 1,3-bis-phosphoglyceric acid undergoes reductive dephosphorylation. So in this second step, what is taking place? Reduction is taking place and dephosphorylation is taking place. Reduction involves reduction of this bis-PGA into 3-PGAL or G3P. Dephosphorylation, that involves removal of one phosphate group, that is from first position. So reduction. So for this reduction, what is required? Protons and electrons are required. These protons and electrons are donated by NADPH and hence NADPH undergoes oxidation into NADP plus by donating protons and electrons to 1,3-bisphosphoglyceric acid, which has been reduced into G3P, that is glyceraldehyde 3-phosphate, or we can call it as 3-phosphoglyceraldehyde. So acid group has been converted into aldehyde by the end of reduction. So reduction occurs by 2 steps. First step is phosphorylation of 3-PGA into 1,3-bis-PGA, during which ATP is consumed. Second step of reduction is reductive dephosphorylation, during which 1,3-bis-PGA undergoes reduction into G3P, and dephosphorylation, during which phosphate present in this bis-PGA, that too at the first position, is removed, and hence only one phosphate is present at third position, and hence it has become G3P, glyceraldehyde 3-phosphate. During reduction, NADPH2H is utilized, whereas during phosphorylation ATP is utilized. Okay, actually for the formation of one molecule of glucose, how many carbon dioxides are utilized? 6 carbon dioxides are utilized. So let's study this cyclic pathway, that is Calvin cycle, by taking 6 carbon dioxides. So 6 carbon dioxides are fixed into 6 molecules of carbon dioxide is fixed into 6 molecules of RUBP, resulting in the formation of 12 molecules of 3-PGA. Now this 12 molecules of 3-PGA undergoes reduction, that is first step of reduction, resulting in the formation of 12 molecules of 1,3-bis-PGA. Now 12 molecules of 1,3-bis-PGA undergoes second step of reduction, that is reductive dephosphorylation to form 12 molecules of G3P or 12 molecules of 3-PGAL, 3-phosphoglyceraldehyde, glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. Okay, now among these 12 molecules, 2 molecules of G3P or trioses or 3-PGAL is transported into cytoplasm of the cell, where these 2 molecules together form one molecule of glucose, that is hexose, 6 carbon compound, that is hexose. So G3P is 3 carbon compound. So 2 molecules of this G3P is transported into cytoplasm of the cell where 1 molecule of glucose is synthesized. That is 6 carbon compound. Okay, now how many molecules are remaining? 10 molecules are remaining. Now these 10 molecules of G3P or 3-PGL undergoes regeneration to form 6 molecules of RUBP. So for the formation of 1 molecule of RUBP, for the regeneration of 1 molecule of RUBP, 1 ATP is consumed. That is in the final step, last step of regeneration. Okay, for the formation of 6 molecules of RUBP, how many ATPs will be consumed? 6 ATPs will be consumed. So, during regeneration, that too in the last step of regeneration, 6 ATPs are consumed. So, 6 ADPs are released. So, this is about the 3 steps involved in Calvin cycle, that is carboxylation, reduction and regeneration. So, this cycle must be continued for 6 times. 6 cycles must be continued, resulting in the formation of 12 molecules of G3P, among which 2 molecules of G3P are transported into cytoplasm for the synthesis of 1 molecule of glucose, and remaining 10 molecules of G3P or trioses are utilized under regeneration. They undergo regeneration to form 6 molecules of RUBP. So, if this cycle is continued for 6 times, then only 1 molecule of glucose is synthesized. Okay. So, what is the energy utilized for the synthesis of 1 molecule of glucose? What is the energy? So, for the synthesis of 1 molecule of glucose, the energy which is utilized is 18 ATPs. 18 ATPs are utilized and 12 NADPH are utilized. 12 NADPH plus H plus are consumed. Among 18 ATP, 12 ATP are consumed. 12 ATP are consumed in the first step of reduction and 6 ATPs are consumed in the final step of regeneration. And among 12 NADPH, all the 12 NADPH are utilized in second step of reduction. That is reductive dephosphorylation. Right. Okay. So, how many carbon dioxides are utilized for the formation of 1 molecule of glucose? 6 carbon dioxides are utilized. Okay. So, for 6 carbon dioxides, for the fixation of 6 carbon dioxides, how many ATP? 18 ATP and 12 NADPH. So, for the fixation of 1 molecule of carbon dioxide, 1 carbon dioxide, how many ATPs and NADPH will be utilized? 3 ATPs and 2 NADPH are utilized. 2 NADPH plus H plus are utilized. So, this is about the brief introduction of Calvin cycle. Now, I want to give you some more Calvin cycle little bit in more detail. Okay, let's study this C3 cycle or Calvin cycle in detail. Okay, so as we discussed now, C3 cycle or Calvin cycle involves 3 steps, that is carboxylation, reduction and regeneration. So, carboxylation first. So, 6 molecules of RUBP undergoes carboxylation into 12 molecules of 3-PGA. So, this RUBP is nothing but ribulose 1,5-bisphosphate, which is 5 carbon compound and it is ketone sugar. So, into this RUBP, that is organic compound, 6 molecules of carbon dioxide is fixed, resulting in the formation of 12 molecules of 3-PGA, and this step is called as carboxylation, and this carboxylation is catalyzed by an enzyme Rubisco. This Rubisco we already studied now. Rubisco is functioning as carboxylase and even oxygenase. So, it is a dual enzyme. It functions as carboxylase and oxygenase depending upon the conditions of the cell, conditions present inside the cell. Okay, right. So, RUBP is 5 carbon compound, 6 molecules of 5 carbon compound. 6 into 5, how many carbons? 30 carbons here. Here, 6 molecules of carbon dioxide. Carbon dioxide is 1 carbon compound, 6 molecules. So, how many carbons are there here? 6 carbons. 6 carbons are fixed into 30 carbons, resulting in the formation of 12 molecules of 3-phosphoglyceric acid. This is 3 carbon compound. 12 molecules. 12 threes are how many carbons? 36 carbons. So, it is balanced. Now, these 12 molecules of 3-phosphoglyceric acid, that is 3 carbon compounds, undergo phosphorylation into 12 molecules of 1,3-bisphosphoglyceric acid. So, this phosphorylation is catalyzed by an enzyme phosphoglycerokinase. So here 1 phosphate is added and hence it has become 3-PGA has become 1,3-bis PGA. Phosphorylation is nothing but addition of phosphate. Where this phosphate is added now? At first position. Already at third position phosphate group is present. Now at first position phosphate group is another phosphate group is added and hence it has become 1,3-bis phosphoglyceric acid. Okay, so this reaction is catalyzed by the enzyme phosphoglycerokinase and here phosphate is donated by ATP and hence 12 molecules of ATP donates 1 phosphate and it has been converted into 12 molecules of ADP, adenosine diphosphate. Okay, now these 12 molecules of 1,3-bis PGA, 1,3-bis phosphoglyceric acid, or we can say diphosphoglyceric acid. 1,3-bis phosphoglyceric acid or diphosphoglyceric acid undergoes reduction to form 12 molecules of G3P, glyceraldehyde 3-phosphate. Acid has been converted into aldehyde, glyceraldehyde 3-phosphate, also called as 3-phosphoglyceraldehyde. It is nothing but triose. So here G3P, glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. That means here phosphate is present at third position. So this reduction, in this reduction what is taking place? Reduction is taking place and even dephosphorylation is taking place and hence it can also be called as reductive dephosphorylation, which is catalyzed by an enzyme phosphoglyceric PGDH I have written, that is nothing but phosphoglycerodehydrogenase. So here dephosphorylation is taking place, reduction is taking place. Dephosphorylation involves the removal of phosphate and even reduction is taking place. For that reduction, protons and electrons are donated by NADPH and hence NADPH is converted into NADP+. So 12 molecules of NADPH are utilized, which will be converted into NADP+ after donating protons and electrons to this compound, that is bis PGA, and hence bis PGA has been dephosphorylated and reduced into G3P, glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. Okay, so here this is 3-carbon compound again. So how many molecules are formed? 12 molecules are formed. Among 12 molecules of G3P or trioses or 3-phosphoglyceraldehyde, 2 molecules are exported into cytoplasm, which are utilized for the synthesis of hexose, that is 1 glucose molecule. Whereas remaining 10 molecules of G3P are utilized for the regeneration of 6 molecules of RUBP. So all these reactions from here are involved in regeneration of 6 molecules of RUBP. Okay, now let's study all these regeneration steps. So among 12 molecules of G3P, that is trioses, 6 molecules of G3P undergo isomerization into 6 molecules of DHAP. 2 molecules of DHAP, here 2 molecules, here 2 molecules. And remaining 6 molecules of G3P remain as G3P only. So these are 2 molecules of G3P, these are 2 molecules of G3P, these are 2 molecules of G3P. So among total 12 molecules of G3P, 6 molecules of G3P is undergoing isomerization into 6 molecules of DHAP and remaining 6 molecules of G3P remains as G3P only, right? Okay, now let's study regeneration steps from here. So here 2 molecules of G3P undergoes condensation with 2 molecules of DHAP. Into what? Into 2 molecules of fructose 1,6-bisphosphate, and this step condensation is catalyzed by an enzyme aldolase. So here 2 molecules of G3P. G3P is 3-carbon compound. 2 molecules means how many carbons are there here? 6 carbons. 6 carbons is reacting with again 6 carbons. DHAP is 3-carbon compound again, which is the isomer of G3P. So here 2 molecules of 3-carbon compound. So here also 6 carbons are there. 6 carbons plus 6 carbons, 12 carbons is formed. So fructose 1,6-bisphosphate is a 6-carbon compound. Here 2 molecules of fructose 1,6-bisphosphate is formed. So 2 molecules into 6 carbons. So 12-carbon compound. 12 carbons is formed. So it is balanced. Okay, now this 2 molecules of fructose 1,6-bisphosphate undergoes dephosphorylation. Dephosphorylation means removal of phosphate. So phosphate is removed, 6-PI is removed. Okay, so dephosphorylation, which is catalyzed by an enzyme fructose 1,6-bisphosphatase, and it results in the formation of 2 molecules of fructose 6-phosphate. So where from this phosphate is removed? Phosphate is removed from first position, and hence it results in the formation of 2 molecules of fructose 6-phosphate, which is again 6-carbon compound. Now this two molecules of G3P. Two molecules of G3P undergoes transketolation with two molecules of fructose 1,6-bisphosphate, resulting in the formation of two compounds. One is erythrose 4-phosphate and the other one is xylulose 5-phosphate. Okay, let's see this reaction. So this transketolation is catalyzed by an enzyme transketolase. So here two molecules of G3P. G3P is 3-carbon compound. So two molecules means 2 into 3, 6 carbons are undergoing transketolation with how many carbons? 12 carbons. Fructose 6-phosphate is 6-carbon compound. Here two molecules are there. So 2 into 6. How many carbons? 12 carbons. So 6 carbons plus 12 carbons, how many? 18 carbons. So 18 carbons must be formed. Let's see whether these 18 carbons are formed or not. So erythrose 4-phosphate, it is 4-carbon compound, 2 molecules. 2 molecules into 4 carbons, how many carbons? 8 carbons here. And xylulose 5-phosphate, this is 5-carbon compound. 2 molecules of 5-carbon compound is formed. 2 into 5, how many carbons here? 10 carbons. So 10 carbons plus 8 carbons together, 18 carbons. So 18 carbons is formed. So balanced. Okay, now these two molecules of erythrose 4-phosphate undergoes condensation with DHAP. DHAP to form what? To form two molecules of pseudoheptulose 1,7-bisphosphate. So this condensation step is catalyzed by aldolase. Now DHAP is 3-carbon compound. Two molecules of 3-carbon compound is nothing but 6 carbons and two molecules of 4-carbon compound is nothing but 8 carbons. So 2 carbons plus 6 carbons, how many carbons now? 14 carbons. Resulting in the formation of 7-carbon compound. Two molecules of 7 carbons. So 2 molecules of 7-carbon compound means 14 carbons is formed. So it is balanced. Okay, now these two molecules of 7-carbon compound, that is pseudoheptulose 1,7-bisphosphate, undergoes dephosphorylation, that is removal of phosphate, where 1 phosphate is removed from first position. So it results in the formation of pseudoheptulose 7-phosphate, two molecules of pseudoheptulose 7-phosphate, which is catalyzed by an enzyme. This reaction, dephosphorylation, is catalyzed by an enzyme pseudoheptulose 1,7-bisphosphatase. Okay, now these remaining two molecules of G3P, undergo transketolation with two molecules of pseudoheptulose 7-phosphate. When it undergoes transketolation, it results in the formation of two compounds. Again, those two compounds, one is ribose 5-phosphate and the other one is xylulose 5-phosphate. Both are 5-carbon compounds. So let's see. So 3-carbon compound, two molecules of 3-carbon compound G3P. That means how many carbons? 6 carbons here. And here how many carbons? Two molecules of 7-carbon compound. That means 14 carbons. So 14 carbons plus 6 carbons, together 20 carbons. 20 carbons resulting in the formation of two compounds. Here two molecules of 5-carbon compound, that is ribose 5-phosphate, 10 carbons. And two molecules of 5-carbon compound is nothing but 10 carbons, that is xylulose 5-phosphate. So 10 carbons plus 10 carbons, 20. So balanced, 20. Right. Now these two molecules of xylulose 5-phosphate and these two molecules of xylulose 5-phosphate, together how many molecules? 4 molecules. 4 molecules of xylulose 5-phosphate undergo epimerization into what? Into 4 molecules of ribulose 5-phosphate. Epimerization is a kind of isomerization only into ribulose 5-phosphate, which is catalyzed by the enzyme ribulose 5-phosphate epimerase. So here 4 molecules of ribulose 5-phosphate is formed, which is 5-carbon compound again. Right. Now these two molecules of ribose 5-phosphate undergo isomerization into 2 molecules of ribulose 5-phosphate, which is catalyzed by an enzyme ribose 5-phosphoisomerase. Okay. Now these 4 molecules of ribulose 5-phosphate and 2 molecules of ribulose 5-phosphate, together how many molecules? 6 molecules. 6 molecules of ribulose 5-phosphate. Together 6 molecules of ribulose 5-phosphate undergo phosphorylation, which is the final step of regeneration. Undergo phosphorylation into what? 6 molecules of RUBP. What is phosphorylation? Addition of phosphate. Where this phosphate is added, new phosphate, it is added at. So what is RUBP? RUBP is ribulose 1,5-bisphosphate. Here phosphate is already present at 5th position, 5th carbon atom. Now the new phosphate is which is added through this step, through this reaction, that is phosphorylation, is added at 1st position, and hence it has become ribulose 1,5-bisphosphate. Ribulose 5-phosphate has become ribulose 1,5-bisphosphate due to the addition of one more phosphate at 1st position. By the reaction phosphorylation phosphorylation which is catalyzed by the enzyme ribulose 5-phosphokinase. 5-phosphokinase, right? And for this phosphorylation, phosphate is donated by ATP. So as there are 6 molecules of ATP is consumed. That means 6 molecules of ATP is donating 6 phosphates and converted into 6 molecules of ADP. Right. So like this, RUBP, 6 molecules of RUBP is regenerated, which is ready to fix again the 6 molecules of carbon dioxide to form what? 12 molecules of 3-PGA, so that this cycle can be continued. Okay, so this is about carboxylation, reduction and regeneration which occurs during Calvin cycle, that is C3 cycle. So carboxylation is one-step reaction, reduction is two-step reaction where phosphorylation and reductive dephosphorylation occurs, and regeneration is 9-step reaction. Before regeneration, 2 molecules of DHAP, that is 2 molecules of G3P which undergoes isomerization into DHAP will be transported into cytoplasm where it will be converted into 1 molecule of hexose, that is 1 molecule of glucose. So for the formation of 1 molecule of glucose, 6 cycles must be continued for the fixation of 6 molecules of carbon dioxide into 6 molecules of RUBP. Right. Here I want to give you one more point. So here 2 molecules of fructose 6-phosphate is formed as an intermediate. Now this fructose 6-phosphate, this molecule, this fructose 6-phosphate can be withdrawn from this Calvin cycle or C3 cycle, and which is utilized as substrate in the process of glycolysis for the release of energy, for the formation of energy. Suppose if the cell is in need of energy, what happens? The intermediate, that is fructose 6-phosphate, which is formed during Calvin cycle or C3 cycle, can be withdrawn from this cycle and it can enter into glycolysis, glycolytic pathway for the release of energy. That is called glycolytic reversal. And I will be explaining about this glycolytic reversal in the next video. You please do watch that next video also to know the details of what is glycolytic reversal. All these steps occurs in reverse. So that is glycolytic reversal. Okay, right. So here I want to give you some more important points. So during this regeneration, many intermediates are formed. Those intermediates are 4 carbon compounds, 5 carbon compounds, 6 carbon compounds and even 7 carbon compounds. So what is the intermediate that is 4 carbon compound? Erythrose 4-phosphate. Erythrose 4-phosphate. How many molecules are formed? 4 molecules are formed. And 5 carbon compound intermediate is xylulose 5-phosphate, ribose 5-phosphate and ribulose 5-phosphate. How many molecules of xylulose 5-phosphate is formed? 4 molecules. Here 2 molecules and here 2 molecules. Xylulose 5-phosphate. So 4 molecules of 5 carbon intermediate is formed, that is xylulose 5-phosphate. Next, 6 carbon intermediate. 6 carbon intermediate is fructose 6-phosphate. How many molecules is formed? 2 molecules. And even fructose 1,6-bisphosphate is also an intermediate. That is also 6 carbon compound. 2 molecules of fructose 1,6-bisphosphate. These are 6 carbon compounds which are the intermediates which are formed during regeneration of RUBP during Calvin cycle. Right. Next, 7 carbon compounds which are the intermediates formed during this regeneration. 7 carbon compounds are pseudoheptulose 1,7-bisphosphate and pseudoheptulose 7-phosphate. How many compounds? 2 compounds. 2 compounds of pseudoheptulose 1,7-bisphosphate and even 2 compounds, 2 molecules. Not 2 compounds, 2 molecules. 2 molecules of pseudoheptulose 7-phosphate. So this is all about intermediates. And even ribulose 5-phosphate is also an intermediate which is 5 carbon compound. How many molecules of ribulose 5-phosphate is formed? 6 molecules of ribulose 5-phosphate. Here 2 molecules and here 2 molecules. And ribose 5-phosphate is also an intermediate which is 5 carbon compound. How many molecules are formed? 2 molecules are formed. So this is about intermediates which are formed during the regeneration of RUBP from how many molecules of G3P? 10 molecules of G3P. So among 12 molecules of G3P, 2 molecules are exported into cytoplasm for the synthesis of 1 molecule of glucose, whereas remaining 10 molecules of G3P are utilized during regeneration. They undergo regeneration into 6 molecules of RUBP. So this is all about different steps, different reactions that are involved in Calvin cycle, that occur during Calvin cycle. Okay, now let's talk about energy. How much amount of energy is utilized for the synthesis of 1 molecule of glucose? So for the synthesis of one molecule of glucose through this Calvin cycle, how many ATP are utilized? 18 ATP are utilized. Among these 18 ATPs, 12 ATPs are utilized in the first step of reduction. 12 ATPs are utilized in the first step of reduction. First step of reduction, how many ATPs? 12 ATPs are utilized. Whereas in the last step of regeneration, how many ATPs are utilized? 6 ATPs are utilized. Next, coming to NADPH. How many NADPH are utilized for the synthesis of glucose? 12 NADPH. Where they are utilized? They are utilized in reduction. What is that? Second step of reduction. So reduction involves two steps. One is phosphorylation and the second one is reductive dephosphorylation. In this reductive dephosphorylation, how many NADPH are utilized? 12 NADPH are utilized. So this is about the energetics of Calvin cycle, that is C3 cycle. In the next video, I will be starting with glycolytic reversal. What is glycolytic reversal? The intermediate, that is fructose 6-phosphate, which is formed during the regeneration of RUBP from G3P, can be withdrawn from this Calvin cycle and it can undergo breakdown through glycolysis for the release of energy if the cell is in need of energy. That is called glycolytic reversal. Hope you understood this concept. Thank you, students.